Hello, I'm Janet Dancy. I'm a medical oncologist and director of the Canadian Cancer Trials Group, and I'm pleased to be given the opportunity to speak to you about cancer drug development and the role of cancer clinical trials. Now, I think people are aware, but it's worth underlining that almost all patients with cancer have longer and better survival now than compared to the 1980s. And some of that improvement in survival and actual reduction in risk of death from cancer is due to lifestyle changes such as cessation of smoking or the use of hormone replacement therapy, better screening and early detection of cancer, but also from improved cancer therapies for the treatment of cancer and the control of cancer-related symptoms. Cancer clinical trials have been critical to generate the evidence of improved outcomes from any cancer treatment. But the process of developing a drug takes considerable time, expertise, and resources, and is often prone to failure. So there's ample opportunity for improvement. So I will be touching on a few things today: where drugs come from, how clinical trials are designed and the endpoints that are used in drug development, the role of industry and academic sponsors of trials, and the lessons learned as we plan to move forward to continue our development of better cancer treatments. In terms of where cancer drugs have been coming from, historically, they've generally been identified by their ability to kill dividing cells. Some of that has been through serendipitous observation, like the discovery that nitrogen mustard shrunk down and eradicated lymph nodes and white blood cells in soldiers who were exposed to nitrogen mustard during the World Wars. Some of it has been through systematic assessment of synthesizing chemicals and testing them in cell lines of cancer for an anti-cancer cell kill effect. These are called empiric screening in cell line models. Some of it has been taking extracts from natural products and then screening them, and when they've shown an anti-cancer effect, then identifying the chemical and synthesizing that chemical. Currently, though, we've moved from what has been called this empiric model of looking at chemicals to see if they kill cells to a more rational or targeted approach, where because we understand better cancer biology, we can identify targets such as a mutated protein and develop chemicals that interact with those targets more specifically, and thus can directly kill cancer cells, preferentially to normal cells. Now, that process of target identification and target validation basically is based on the fact that a target must be present in cancer, it contributes to the growth of cancer, and it can be engaged by a drug for an anti-cancer effect. Drug development begins with a research and development phase where the target is identified and qualified. Target identification is generating that evidence that the target is manipulatable in a way that can have that anti-cancer effect. And target qualification is systematically going through a series of preclinical and perhaps even limited clinical studies to establish the scientific validity of the agent that is being targeted by that drug. So that process might involve discovering a biomolecule of interest, a mutated protein, for example, in an important pathway that contributes to cancer cell growth, evaluating its potential as a target, designing a way of assessing the effect of that target when it is modulated by a chemical with an anti-cancer effect, so that there can be a construction of a high throughput screen that can subsequently have tens of thousands of chemicals run through that high throughput screen to start identifying those that interact with the target and have that anti-target or that proposed target modulatory effect. Those are called hits. And then the process involves evaluating the hits. This phase, the key deliverables are a known and qualified target, the disease-relevant experimental models for subsequent evaluation, and a set of initial compounds that can be further refined. The preclinical development phase continues with improving those initial chemicals and turning them into a clinical candidate, a potential drug. And that process involves a lot of chemistry to improve the target specificity, improve the pharmacology of the drug to make it more of a drug that you can administer safely and effectively in patients, proven its efficacy in preclinical models, proving its safety by assessing formally toxicity in preclinical models, usually 2 animal models, and developing a process to manufacture the product to a clinical grade to administer to people. The key deliverables in this phase is you have a candidate drug for clinical testing. It's relatively specific for the target, has few off-target normal tissue effects, has the properties of a drug, meaning that it can be administered to people, can be taken up, it can get to the cell of interest, it can get to the target of interest and have that target effect, on-target effect. And it can be safely administered to patients. Once those preclinical steps are done, a drug enters clinical development, starting a series of phases of evaluation, beginning in phase 1, where the question is the drug safe, and what is a safe dose and schedule to administer it? Phase 1 trials usually start at a low dose and progressively escalate the dose until we get into a range where we anticipate an anti-cancer effect and or there may be side effects that limit further dose escalation. Once a safe dose and schedule is identified, it moves to phase 2, where there's additional assessment of safety, but we also start looking for an anti-cancer effect. And if that anti-cancer effect is promising and toxicity is absent or tolerable, the drug progresses to phase 3, where it is compared to standard of care, and we determine whether it works better than standard of care. If it does, it has the potential to become marketable and administered to patients as a treatment option. So phase 1 trials are small, single arm. They begin with dose escalation, establish safe dose and schedule. Phase 2 trials are generally small, usually less than 100 patients, may have a single arm or multiple arms, may have a comparator, a control arm. But phase 3 trials are the largest trials. They usually always have a comparator arm, a standard of care. They're randomized. They may have a placebo, not as a substitute for standard of care, but in addition to standard of care. And they're designed such that if they are positive, they can potentially receive regulatory approval as a new standard of care. Now, why is randomization so important for phase 3 trials to definitively define clinical benefit against standard of care? Well, it's because we need to control for certain types of biases and confounding effects. It is correct that we may be able to identify which patients may do better or worse. And if we preferentially selected patients who would do better for one treatment versus another, we bias the result quite independently of the actual treatment effect. Similarly, there may be confounding factors, things that we don't know or don't quite understand that may be imbalanced between treatment groups if we were selecting treatments for patients in a non-randomized fashion. Randomization is meant to balance confounding effects that may influence outcomes and to control and mitigate and reduce selection bias that may also confound outcomes. We use placebos as a way of controlling for the assessment of endpoints that are more subjective rather than objectively measured. So a placebo is a substance that is made to look like the experimental treatment but has no therapeutic effect. And it again is used in situations to control for bias. In this case, informational bias, i.e., the assessment that we're doing is something that may be influenced by knowledge of whether the patient is on the experimental treatment or not. And a classic example of when we might use a placebo is when we're assessing a patient-reported outcome, such as a symptom improvement, where the improvement in symptoms may be related to whether a patient knows, or the doctor knows, if the patient is on an experimental drug versus not. So it's important also, I think, to touch on who can participate in trials and how trials are conducted. All clinical trials have a protocol, which is essentially a very detailed instruction manual for everybody involved in the study that explains who can be enrolled, how they are to be managed on the trial, how the assessments are to be done, how the treatment's to be administered, how that treatment might be changed if patients had side effects. So not everybody with a cancer can go on a trial. They have to meet certain eligibility criteria related to the type of cancer, the stage of the cancer, other potential features, their other health factors that they may have. But patients who are eligible may be approached, in which case the trial, the purpose, the activities on the trial, the risks, the benefits, the options are explained to them in detail through an informed consent document. And if patients who go through the informed consent document, go through the information, ask questions, are satisfied and interested in participation, they can be enrolled on the trial. There are many different groups involved to ensure the safety and privacy and the rights of patients on trials. This starts with the review of the protocol and the research proposal. It includes the research ethics boards at individual sites that may be participating in the trials. All participants must follow good clinical practice standards and have to demonstrate training on good clinical practice principles. They have to follow privacy regulations. All large phase 3 trials have to have an independent data safety monitoring committee, an independent group who evaluate the data at particular intervals to determine whether it's safe for the trial to continue or whether it should be stopped early. And national regulatory agencies like Health Canada or the US Food and Drug Administration have oversight responsibilities on all trials of investigational drugs. So drug trials basically have to be filed with these agencies to proceed and reported on to them over the course of the trial. Now, clinical trials transition through these phases based on whether they meet their primary outcomes and key secondary outcomes. So in early phase trials, these focus on safety, establishing a dose and schedule, and seeing shrinkage of tumor. In later phase trials, it may be delayed tumor growth, longer life, fewer side effects, better overall survival. Regulatory agencies assess for safety and efficacy, but also the quality of the evidence and the manufacturing quality of the product in thinking through their decisions about whether to approve a drug for marketing purposes. Now, the requirements, the benchmark that has to be met for efficacy and safety are context dependent. It depends on the patient, the disease setting, the availability of alternative treatments, the overall safety profile. Regulatory agencies do not assess cost or value. The cost might actually not be known at that point. The assumption is that regulatory authorities have authority based on legislation that focuses on efficacy and safety evaluation. Cost and value are determined by market forces, third-party payers, public payers, patients, clinicians who make decisions about whether this treatment is worth administering in particular situations. The overall process takes several years, and it's not particularly efficient. Estimates suggest that the overall clinical development success rate from the time of preclinical evaluation forward is about 5%. And the actual cost of developing a drug is about $1 billion. The causes of the slow development and the risk of failure are multiple. Cancer is a complex disease. Druggable targets are few. Cancer treatment is complex. There are more effective treatments, and so each new drug has to beat standard of care, means the bar for efficacy continues to go up. The causes of delays have more to do with the size of the trial. and the duration required to be able to get to the analysis point. Few patients participate in trials. Estimate is about 5% in North America. The endpoints may occur over long durations, may take years before a trial is ready for analysis. And the cancer clinical trials, however, are evolving to reduce the time to market and to improve the success rate. Right now, it's really dependent on knowing you have the right drug dose schedule, the right patient and disease, the right trial design and endpoints. And so improving that assessment to be certain that you do have those features will definitely improve the clinical trial outcome. So in terms of what's changing, first of all, in the preclinical development, there's greater rigor, greater understanding of cancer biology, greater more rigorous approach to target selection. There's also improved chemistry and manufacturing. So there is a whole range of type of drug classes now to interact with targets. In addition to traditional chemotherapy, we have small molecules that are able to target tumor or cancer-specific targets. We have immunotherapies, we have proteins, mostly antibodies or conjugated antibodies that can be highly effective in interacting with targets and killing cancer cells. We have more efficient trial designs, particularly adaptive trial designs, Bayesian designs. There is a move towards earlier endpoints, which is controversial. You'll hear more about that. We're looking for bigger effects, bigger steps forward in terms of improvement in patient benefit. And we're trying to broaden eligibility criteria to improve patient access and participation in trials. There's an overall move to improve trial operational efficiency and trial data management. Now, the endpoints of clinical trials clearly are the key determinants of progression through clinical development and the overall success of the clinical trial in its attempt to get to market. If you look at the Food and Drug Administration's clinical trial endpoints for approval of cancer drugs and biologics guidance, you can see that there are a whole list of cancer endpoints for clinical trials that may lead to traditional approval or what's called accelerated approval for patients who have incurable diseases and there's a need to identify new treatments. These endpoints include overall survival, but also symptom improvement. These are both considered the key clinical benefit endpoints. Other endpoints might be delayed progression, delayed relapse, tumor regression, especially complete response, or delayed progression, that delay of tumor growth. These have been used for traditional or accelerated approval in specific settings, and these settings are clearly context dependent. The magnitude of the effect, the duration of the effect, the availability of therapy, the disease setting, whether it's incurable disease with no effective treatments, and the toxicity of the treatment all factor into whether that threshold for regulatory approval is considered to have been met. Now, the benefits of treatment generally focus on patients living longer or their cancer symptoms are improved or controlled. The toxicity is managed and overall patients are doing better. Those thresholds really do depend on magnitude and benefit. On the right side is an attempt to illustrate that if tumor growth occurs relatively quickly, even if it occurs after there's been some evidence of tumor shrinkage, it probably isn't really conferring much of a clinical benefit. But a delayed in tumor growth may be a surrogate for a clinical benefit if symptoms are improved or cancer symptoms are delayed, or if patients not experiencing significant side effects of treatment, they may be doing better on treatment than they would alternatively. And that period where they're doing better may be longer compared to the previous treatment. It's difficult to show that, though, that patients are doing better because we're not, often in the context of trials, we haven't been measuring the key comparison, which may be how people are doing on the experimental treatment without that tumor delay, based and compared to what happened to those patients who had a shorter period, had progressed on standard treatment, and are now in the next phase of their own illness where their tumor is worse. They may be having symptoms. They may be receiving a more toxic therapy. We actually haven't evaluated quality of life, toxicity in that period of time after patients have progressed in most clinical trials. Now, the key determinants of the size of a trial, which obviously lends itself to an increased complexity, increased duration, relate to what is the primary endpoint, what is the disease setting, what's the statistical analysis approach, what are the results with standard treatment, what's the size of the treatment difference and the degree of certainty to be able to detect that difference built into the trial, and the completeness of the data collection, because missing data actually impacts on the sample size required to show a treatment effect. Just as an illustration, I've described a trial published in the New England Journal that was designed to show or to be able to detect a difference of 33% risk of progression of an experimental treatment versus control. So that 33% reduction in risk of progression, or 50% improvement in delay in progression from 6 months to 9 months, required almost 400 patients to show a difference. That number would increase if, for example, you wanted to show a smaller difference. That number might also increase if the progression-free survival in the control arm wasn't 6 months, but 18 months, and you were still trying to show a difference of 3 months in that setting. The sample size would be considerably larger. It would also take a longer period of time, because it would take a median of at least 18 months versus 6 months for the events, for 50% of the events to occur. Now, there are times where cancer drugs appear to show early signals of really impressive anti-cancer effect. They show deep responses, and those responses are durable in those early phase trials. And when this happens, there's an ongoing discussion about whether such effects really require large randomized phase 3 trials before approval. In one facetious article published in the BMJ in 2003, those big effects from treatments were compared to parachutes, where we haven't done randomized controlled trials of parachutes versus no parachutes to prevent death from jumping out of a plane. It's not a completely accurate analogy for the most part, but it is a way of illustrating the challenge sometimes for patients who have incurable disease, very few, if any, medical options to control that disease. And then there's the potential from an early phase trial of a signal of an anti-cancer effect that might be a benefit to them. Trial efficiency around design elements have increasingly focused on adaptive trials, where trials basically blend phase 1 and phase 2 and sometimes phase 3 all within the same protocol. Each step is clearly defined within the protocol, but the overall protocol becomes quite complex, which is suggested on the schematic on the right side, which describes a clinical trial of a immunotherapy that was conducted. However, they do reduce the interval between the phases required for trial startup as each new trial phase progressed, and so are becoming quite common now in cancer clinical trials. We're also moving towards removing barriers to patient participation, broadening eligibility criteria, improving access by reducing geographic barriers, providing culturally appropriate information on trials, different languages, etc. And compensation for trials, travel, decentralizing trial activities. All of these are meant to try to improve, if you like, the interest and ability of patients to participate if they're interested in participating. Now, obviously, industry is the major driver of bringing new drugs to market, but they aren't the only groups who are interested in doing clinical trials, testing new drugs, and defining new areas where those drugs may be effective for cancer patients. Academic-led clinical trial organizations like the Cancer Clinical Trial Group also are capable and have a long history of conducting the full range of cancer clinical trials, including phase 3 trials that define new standards of care and can lead to marketing approval. And in many ways, the academic trial groups like ourselves are the ones who are most focused on both defining the true spectrum of benefit, evaluating them in patient populations that are more representative of the real world, not so highly restricted eligibility criteria. And they're also ones that are more likely to design the trial in a way and in a setting that's most relevant to our Canadian population. So this next few slides are some shameless self-promotion on behalf of the CTG, which is a cooperative group that has been designing and conducting clinical trials for about 40 years now. It's a national program of the Canadian Cancer Society. Its headquarters is based here at Queen's University, but it engages the scientific leaders across the country, and it has a network that pretty much spans every cancer center and every hospital in Canada interested and capable of conducting cancer clinical trials, as demonstrated on this slide. We are linked to peer groups across the world, in the United States, in Europe, in Australia and China, who are similarly interested in developing and conducting these types of trials. Sometimes they lead trials that we join. Sometimes we lead trials that they join. And collectively, we expand the number of clinical trials, the number of opportunities for patients to have access to treatments on trials, and defining potentially new standards of care for future patients. So we are the largest network in Canada. We have conducted over 600 trials involving over 100,000 patients. We currently have about 150 trials running across the full spectrum of trials. And we also have a tumor bank that is able to collect and manage samples from patients on the trials for additional research. To date, we've pretty much conducted trials that have identified treatments for patients with all of these types of cancers. And some of the trials that we've led that have changed standards of care include defining new standards of care for breast cancer patients, lung cancer patients, patients with brain tumors, bowel tumors, blood tumors, in supportive care trials to control pain. The CTG has many firsts as a research institute in Canada. We are the first and largest cancer research network, the only cancer research network that has led trials that have directly defined new drug approvals worldwide. The first and only non-U.S. member of the U.S. National Cancer Network. We're funded from the National Institutes of Health in the United States to conduct cancer trials. And among the trials that we've conducted, 2 were reported at the largest cancer meeting as the top 2 abstracts in 2016, demonstrating the quality and the impact of our work. And we do this because of donors to the Canadian Cancer Society. Our program grant is the 5-year grant. It's the largest grant from the Society, and we use that money and leverage it to acquire additional grants from public sources and from industry to conduct trials that we sponsor. So we leverage dollars up to 7-10-fold at times to be able to do our studies. So we have proven success. We have a great network of investigators, a track record for scientific and operational excellence and data quality, and we've been a great partner with the Canadian Cancer Society and others. So cancer drug development is failure prone, slow, and expensive, but it has led to major improvements in outcomes for cancer, especially in recent years. The reasons for those failures, they're complicated. They're related to the complexity of cancer, the ability to identify druggable targets, and the fact that new treatments have to beat best standards of care, and that relatively few patients are able to participate in trials, and endpoints occur after long durations. So the process is slow and the process is expensive, and it may fail. However, the process is also evolving, so that we have better drugs to test, more efficient designs, and we are able to reduce, hopefully, the time for effective therapies to get to patients. So thank you very much for your attention. I wish I were there to answer questions, but I know the other experts in the room can likely address them in my absence. So take care.